Hello, my name is Pierre Massion. I'm a professor of medicine and cancer biology here at Vanderbilt. And it's my pleasure to introduce you to a particular topic of research interest in my laboratory. I'm a physician scientist, and I spend about 75% of my time to understand the molecular underpinnings of lung cancer development. As you know, lung cancer is a big problem. It's the number one cancer killer in the nation and worldwide. It kills actually more people than colon, breast, and prostate cancer combined. And it kills twice as many women than breast cancer, a fact that most women are not quite aware of. So this is a big problem. One of the biggest problems to tackle lung cancer is actually to detect it early. What's the difficulty about detecting lung cancer early is that people don't complain about a nodule or a cancer growing in their lungs. If that's the case, we have to find strategies to eventually screen some of these people. And we succeeded doing this. Since 2011 now, we have a method to screen people at high risk for lung cancer. It's called low-dose chest CT. So we select people that are high risk, and specifically people who are older than 55 and who have smoked at least a pack a day for 30 years, and people who eventually undergo these programs of screening have a 20% chance to reduce their lung cancer specific mortality. These results have really prompted a large screening effort across the nation and maybe will be expanded to other nations as well. At the moment, although the screening programs are just opening, we are really aiming for detecting the disease early. Why? Because the facts are, should you consider the entire lung cancer population, only about 17% will be alive at 5 years. But when you think about the early detected lung cancers, the stage 1 lung cancers, 75% of them should be expected to be alive at 5 years, a great difference. So the hope is actually with low-dose chest CT, we'll diagnose lung cancer early. But we also realize that chest CT in high-risk individuals is not a panacea. We need actually more and better tests to really get to these lesions early. Early because at that time you can cut them out, and surgery is the best treatment for lung cancer. Now, if you think about the entire U.S. population at high risk for lung cancer and you were to take them in a screening program, that'd be great, but you would still solve only about 10% of all these patients with lung cancer. So we need better tests. Our laboratory has been very interested in looking at what's happening in the airway epithelium. In fact, this is where the problem is happening, in the airway epithelium. And the airway epithelium is a very large surface of cells that are happily living and eventually acquiring a mutation. And let me bring you to this particular topic. What I'm going to try to show you in 3 slides is that we interrogated what's happening early in the airway epithelium when it is exposed to tobacco smoke primarily. And we ask what are the proteins that are altered and how does that eventually confer a risk for developing lung cancer. So here's the concept of the field of cancerization. What do we mean? Well, we mean that as people smoke, they expose the entire airway epithelium to tobacco smoke. And if you look at your airway epithelium and you lay it flat on a surface, it covers the surface of a tennis court. Now, if you smoke sufficiently, the tobacco will cause the tennis court to turn gray and eventually to acquire a series of mutations in the DNA of all these cells in a stochastic manner. It's totally random, and we really don't find in most high-risk individuals evidence for any cancers as of yet. But one day, eventually, in about 15 to 20% of these high-risk folks, cancer will develop. And here it comes. At the top left corner of this tennis court, the cancer has developed. So one of our biggest challenge in early detection of lung cancer is to find these lesions early or find risk predictors of developing this disease. And so we need to access the lungs somehow. How would you want to access the lungs? Well, one way is to eventually biopsy an area of the field of interest. think that we could take a piece of this tennis court and ask whether it tells us whether you're high risk for developing lung cancer. This is the research we undertook in the laboratory. And here is what we did. We asked who is really at high risk for developing lung cancer, and we took a proteomic analysis approach for this. We took the airway epithelium of people at high risk, at moderate risk, and no risk or low risk, and we obtained brushings for the central airway epithelium, as you see in this figure at the top left corner. We looked at these airway epithelial cells, we digested them with trypsin, obtained a peptide soup, if you wish, and analyzed it by shotgun proteomics by tandem mass spectrometry. Identified the proteins of interest and asked what were the differences between the proteins in the low risk, moderate risk, or high risk group. And what we found about 350 different proteins that were upregulated with risk increase. What were those proteins? Well, they're very much enriched for what we know now as carbohydrate metabolism-related proteins. This slide pictures a series of metabolic pathways, including glycogenesis and glycolysis, the TCA cycle. But the point of this slide is to show you that we identified the proteins that are labeled in blue and in red, but those in red were upregulated with risk. And these are, for the most part, key regulator of metabolism in the glycolytic pathway, in the pentose phosphate pathway, and also in the glutamine pathway dependence. So we were able to then validate these experiments with orthogonal methods, and also in a cell culture system where we expose airway epithelial cells to tobacco smoke condensate. After months of exposure, the cells start developing this metabolic reprogramming, and we find many of these proteins labeled in red here being upregulated in the cell culture condition. So this was quite exciting to us. And then we asked, so what's the relevance of this? Well, when you look at the slide, we found that the glutamine metabolism, in addition to the glycolytic pathway, was quite upregulated. And it's upregulated in addition to the expression of a series of glutamine transporters. So the question now is, what's happening in these cells that are undergoing more glycolysis? They are consuming more lactate, producing more lactate, and consuming quite a bit of glutamine. Can we deprive them from glutamine? How do we understand the transport of glutamine in the cells, and how is glutamine uptaken? Can we use these glutamine transporters that are pictured at the surface of the cell with these pyramidal diagrams? And particularly, we've been focusing on LCC1A5 and XCT. These two proteins can be targeted actually for molecular imaging. Quite exciting, because we could bring this molecular imaging to eventually an early detection method using, for example, the PET-CT technology. So what did I tell you about? I told you about a story where we are trying to investigate the determinants of lung cancer development in high-risk individuals. We discovered that metabolic reprogramming was happening early, before the cancer stage. And now we have a series of unanswered questions. First, we need to characterize this a lot better, determine what are the key regulator of this metabolic reprogramming, its dose dependency, and very importantly, how does this metabolic reprogramming could be eventually associated with one of the key determinants of tumor development, which is actually development of genomic instability in the cell, where a large number of mutations are acquired, chromosomal segregation becomes abnormal, chromosome breaks from one another, and the cell becomes quite unstable and eventually turns into malignancy. So our laboratory at the moment is really very much focusing on understanding the relationship between metabolic reprogramming on one hand and pushing the cells to become genomically unstable. So I thank you for your attention, and feel free to contact me should you be interested. My email is pierre.massion@vanderbilt.edu. Thank you for your attention.